Good afternoon, everyone, and welcome to GenQA's Focus On webinar. We have a great pleasure to introduce one of our guest speakers today, who has been a real influence and a great supporter of GenQA's activities over many, many years now, and that's Ian Berry, who's a consultant clinical scientist and the rare disease lead at the Southwest Genomic Laboratory Hub in the UK. We're also joined today by two of my colleagues, or three of my colleagues, who are all deputy directors in GenQA. We have Farah.
Hi.
Dave.
Hi, afternoon, everyone.
And Melody.
Hello.
And all three are involved in managing and providing external quality assessments for rare and inherited disorders. So I'm Sandy Deans and I'm Director of GenQA, and as I say, it's a great pleasure to introduce Ian. So Ian, I'm going to hand over to you now, and would appreciate if you give us your views and an update on the principles for efficient and proportionate analysis of genomic variants.
Thank you, Sandy, and thank you for the invitation to join you today. So if we can move on to the next slide, I can tell you a little bit about what I'm hoping to cover today. So I think essentially the context of the majority of what I'm talking about relates really primarily to sequencing technologies and how we can deliver the results and the outcomes of those tests on rare disease patients in as streamlined and efficient way as possible. But it will include some content relating to sort of tangentially related areas, such as the analysis of large copy number variants on whole genome sequencing. And I'm really aiming to cover the points that are noted below. So primarily the efficient and proportionate assessment of sequence variants. I'm going to talk a little bit about some of the real key resources to get the annotations that you need to make those variant analyses as efficient as possible, particularly talking about the DECIPER interface, which is based in the UK, and various other tools. Going to talk a little bit about the appropriate recording and reporting of results from these technologies, the effective assessment of copy number variants from whole genome sequencing and NGS assays, and also talk a little bit about the assessment of sensitivity and quality in genomic assays, so particularly focusing on coverage and other associated aspects of that picture. So next slide, please. I think really what this boils down to, and the key issue that this boils down to for many laboratories, is how we balance risk. And I think probably fair to say in clinical genomic science, we're very good at the left-hand side aspects of this slide. So thinking about how the quality of our tests, thinking about the sensitivity of our tests, thinking about ensuring that we don't miss diagnoses, that we review variants in a comprehensive and effective way, and also ensuring that our analysis scope is sufficient to maximise the diagnosis, that we have good coverage of our assays, that our variant filtering is appropriate, that our CNV and structural variant detection is in there. But what we're not always great at, I don't think, is thinking about the risks on the right-hand side of this balance here, to ensure that we're doing things in an efficient way, reporting our cases in a clinically useful time frame, and that we're delivering our workload with a financially sustainable approach and a financially sustainable workforce. And there are significant risks there, clinical risks as well as operational risks on that side of the picture, in terms of things like patients dying of preventable disorders because they're waiting for a result, patients waiting to make reproductive choices based on a test that's potentially taking a long time. And I think really sometimes we need to redress that balance and ensure that we're doing the stuff on the left-hand side, but also on the right-hand side. And the key aspect of that, I think, is prioritizing the activities that add value to our analyses, and that's what I'm going to be talking about today. We could move on to the next slide. So by way of context, really, I wanted to put up this little questionnaire here, and this was a group of us in the Association of Clinical Genomic Science got together in London last year to discuss the aspects of delivery of service that would ensure that we have efficient and sort of financially sustainable services in our region. This was a question that was asked to the group, and the people were allowed to anonymously answer: what's the appropriate period of time for a scientist, a competent analyst, to spend on an average for a large panel test or a whole genome sequencing test, just considering the analytical aspects, so not the upstream aspects of dealing with the referral, the technical time, the pipeline time, or the reporting time downstream. And as you see here, the majority of respondents thought somewhere between 30 and 90 minutes is appropriate for that. And actually, you sort of see the problem here, because if you extrapolate that out, that is assuming that a competent analyst is able to deliver somewhere between 5 and 15 WGS or NGS analyses in a typical working day. And I think it's probably fair to say in most labs that's not actually the case. So really this is aspirational rather than a picture of where we're at. And I suppose part of what I want to talk to today is to think about how we can get closer to achieving that aspiration. Could we move on to the next slide, please? And I think that probably the main challenge we face with these analyses is the fact that large panels, whole exome sequencing and WGS generate very large numbers of variants, and that even relatively streamlined pipelines that deliver small numbers of variants back to analysts will still generally give us a fairly significant number of variants for a manual analyst to pass through. And nearly all of these will have very little clinical value or no clinical value at all. They'll be benign, lightly benign or weak VUSs. I think probably all attendees would agree that a full workup using the ACMG variant assessment guidelines is quite time consuming, and I would argue that it's inefficient and unnecessary. So in our laboratory, we focused on developing a series of simple criteria that are really embedded in the ACMG guidelines and in the principles of the 2024 ACGS best practice guidelines for variant classification, that will allow us to identify these low clinical value variants and record their reasons for dismissal without an extensive workup, potentially doing it in really a few seconds of time for the majority of variants. And there are 5 sort of key criteria around that will allow us to exclude our variants without doing a more formal classification when one or more of them applies. I won't labour the point on those on this particular slide, as I'll be going into those in more detail later, but I have listed them here for you to see. And I think a key principle really, again, taken out of the 2024 best practice guidelines, is that in most clinical settings and for most genes or regions, variants of uncertain significance and with a low level of supportive evidence are almost invariably unlikely to be disease causing and should only be reported in exceptional circumstances. So we're happy, I think, from our guidance, at least in the UK, that these variants are not ones that we should be spending a great deal of time on, and they're not ones that are adding value to our analysis. So next slide, please. So sort of pause the flow a little bit, just throw in a small plug here. So I'm going to be talking a little bit about DECIPER, which I'm sure, again, many of the attendees will be familiar with, and the use of that certainly for answering some of those key criteria questions that I'm talking about in terms of being able to dismiss a variant efficiently. It's a great tool, I think, and I'm quite evangelical about it. We certainly use it a great deal in my lab, and I think one of the really useful pieces of functionality with DECIPER is that you're able to create your variant very quickly, either by a URL, as you can see here, which can be sort of pinged off from your decision support tool or your pipeline output from your NGS or WGS or even your microarray analysis, or it can be searched for very quickly in the DECIPER search bar. So you can create your variant and then answer most of those questions that you need to define whether the answer to your key criteria, the 5 criteria that I've listed down there, is yes or no. Of course, there are other resources available, and if you've got a really good decision support tool, or you've got a bespoke laboratory variant triage software that provides those annotations, then by all means do use those. But DECIPER is a good tool, and that it's free, readily accessible, and I think pretty much everybody can get to grips with it very quickly and very effectively. So next slide, please. So the first of the 5 criteria that I'm going to talk about in a bit more detail, which would allow us to get past our variant without doing a significant workup, and potentially in many cases to dismiss a variant in very quick time, would be variants where the phenotype is clearly incompatible with the clinical presentation of the patient. Now, I think there are some assumptions at play here around what is reasonable and what it's not reasonable to expect a clinical scientist to be able to do in terms of making an association with a phenotype. Now, I would say that absolutely clinical scientists shouldn't be expected to infer particularly complex phenotypic fits in esoteric and specialist clinical areas. So I wouldn't expect clinical scientists, for example, to be going through a neuroradiological MRI report and being able to determine whether or not a gene fits with the esoteric features from that, or necessarily to be able to imply a phenotypic fit in complex dysmorphology. And indeed, when we have referrals from mainstream pediatrics, that by necessity, in many cases, the dysmorphology workup won't necessarily be that great. But I would, however, expect that clinical scientists should be able to distinguish these clearly non-overlapping or binary phenotypes whereby they absolutely don't fit with the gene in question. So the example they're given here would be a skeletal dysplasia gene in a patient with an intellectual disability phenotype, or essentially non-overlapping phenotypes such as micro and macrocephaly. The majority of ACMG criteria, as I'm sure you're aware, aren't applicable if the phenotype's not compatible with the gene. And I think this is increasingly becoming an important factor. 10 years ago, most of us were doing relatively small panels that were relatively phenotypically specific, and actually the majority of genes on those panels would be compatible with the patient's phenotype. Now, of course, we're doing a lot more gene agnostic work, exome trios, genome trios, very large panels, such as the R27 pediatric disorders panel, which is applied in England on whole genome sequencing with 2,500 genes on it. By necessity, in those panels, of course, many of those genes don't fit the patient's phenotype in any way. I think another important point to note regarding this area is that discarding a variant at the phenotype assessment stage is a really good way to mitigate against incidental findings. There are many genes, of course, that will be present in pediatric disorder panels, which can also cause later onset adult conditions that are not related to the patient's phenotype. So I suppose examples would be the BRCA1 and BRCA2 genes, potentially associated with Fanconi anemia, which you could have returned as an incidental finding in a pediatric panel. But if, of course, you pick those genes up and the first thing you do is look at the phenotype, that's the first annotation you assess, then you can throw those out at that stage in many cases for a patient who does not have a Fanconi anemia compatible phenotype, and therefore mitigate the risk of giving yourself a lot of extra work and potentially doing patient harm in terms of uncovering an incidental finding. So next slide, please. So I think there's a few elements of caution to note around scientists doing a phenotypic assessment on a variant, and that we need to be cautious that there are many phenotypes and many clinical scenarios where those phenotypes potentially won't have been assessed and certainly won't be disclosed by clinicians. Many mainstream clinicians who are specialists in a certain area are not going to look at the holistic picture of the patient and potentially aren't going to reliably investigate the patient's family history. So you end up with a situation here whereby you have your scientists looking at different parts of this elephant and not putting the whole picture together because they're only looking at one bit of it. So an example of that in practice would be that a neurologist is highly unlikely to investigate a patient's renal history. Likewise, a nephrologist highly unlikely to investigate a patient's ophthalmological history. So really the emphasis here has to be very much on non-overlapping or completely incompatible phenotypes when they are declared on the referral paperwork for your patient. And of course, this doesn't need to be a standalone criterion. If we're unsure and we think that the phenotype is probably incompatible with a patient, we can do some very, very quick checks and cursory checks for red flags that would potentially flag a variant up as pathogenic. We can look at the frequency on NOMAD, whether the variant's been reported to ClinVar or not in a tool such as DECIPER. And if we think that the phenotype's unlikely and there aren't any other red flags that indicate pathogenicity, then the overwhelming likelihood is that variant will be irrelevant. So next slide, please. Again, going back to where we pull this data, I think the gene clinical tab on DECIPER is a great one-stop shop for this. We can look at the different, the key curation sources for gene disease associations. So we have here the GenFan resource. We have OMIM, which is I think the resource that we're typically the most familiar with in the UK. But certainly one that may not update that quickly with new disease associations that are published. But there's also gene reviews and the ClinGen curated assessments as well that give you definitive, moderate, limited, etc. gene disease association curation. So great resource really to see all the phenotypic spectrum of a gene, and also the predictive scores there that will indicate whether a gene is likely caused, a gene is likely constrained against loss of function variation or not, and also a link out to the Genomics England Panel App tool. So really great sort of one-stop shop for assessing what phenotypes are associated with a gene. Next slide, please. So moving on from phenotypic assessments to allele frequency, which I think is one of the key items for being able to dismiss variants relatively quickly. And obviously we're aware that the ACMG criteria BS1 is likely to be applicable for a variant where the frequency in the general population, so in a tool like Nomad, for example, is too high to be compatible with pathogenicity in that gene. And in all cases, that would render a variant at best VUS and probably more likely benign or lightly benign. And I'd say this is often intuitively quite obvious in many contexts, and that we can use our scientific judgment as trained scientists to assess this without having to do a lot of calculations. So examples would be a rare dominant disease where we've got many heterozygotes apparent in Nomad, or a rare recessive disease with multiple homozygotes, or even considering a disorder where the gene disease association is very much for a very severe early onset condition. If you have any heterozygotes at all in a dominant context in Nomad, that likely is going to exclude pathogenicity, or at least make it very unlikely. So we see the DECIPER annotation and allele frequency tab on the right hand side here. So this is a great sort of at a glance digestible first port of call here. This gives you the data from Nomad version 2 and version 3 put together, which is great. It's both exome and genome data, the vast majority of it from exomes, and covering around 400,000 alleles for coding regions in the genome that are readily exomable. But what we may find is that variants may not be readily sort of dismissible from that data, and we may want to go to a larger disease cohort, a larger population cohort rather, which would require us to go to Nomad version 4 using the online browser tool. Again, if you have Nomad version 4 annotation within your pipeline, all the better. But I quite like personally taking the GRCH 38 coordinate from the variant page on DECIPER, shown here in the red box, and putting that into the search tool in Nomad, because that actually gives you the region rather than just the specific variant information in Nomad. And you can see if, for example, you have a missense variant, as we can see here in this gene CHD4 affecting arginine 645. We can also see if there are other missense variants in Nomad at the same position, which might make intuitively a missense variant potentially slightly less likely to be pathogenic. Also worth noting that Nomad version 4 has around 4 times as much data, 4 times as many alleles as version 2 and version 3, and the majority of these are from the Northern European extraction, mostly UK based and mostly from the UK Biobank. So certainly in the hands of our UK based lab, we've seen many variants that have got 0 frequency in Nomad version 2, version 3, which may have 20 or more heterozygotes in Nomad version 4. So useful tool if the version 2 and version 3 data doesn't get you to where you need to be. So next slide, please. So I think it's fair to say that sometimes the Nomad frequency will not be an intuitive, easy stop for you to say this variant is clearly non-pathogenic, and where it isn't obvious, you can potentially use it as a supporting feature alongside other factors to dismiss your variant or to suggest that it's non-pathogenic. So examples would be looking for an entry in ClinVar if it's a VUS or lower. And if it's not in an obvious hotspot region of the gene, we may at that point decide that the likelihood of this variant being pathogenic is extremely low and that we can move on without further work. So a good example here, PTPN11, this gene is associated with Noonan syndrome. We have a missense variant in this gene affecting position tyrosine 197. We can see from the Nomad version 2 and version 3 data, there are 2 individuals in Nomad with this variant. That wouldn't exclude pathogenicity in Noonan syndrome, as it's variably expressive, and there are many individuals with PTPN11 pathogenic variants in Nomad. But if we also use the protein genomic tool on DECIPER. We can see here a really nice schematic in the bottom right-hand corner of the gene. We can see where our variant is with the vertical yellow line, and we can see that it's not within any of the functional domains of this protein, and it's not within any of the clear missense hotspots which are associated with Noonan syndrome in this disorder. We can furthermore see the variant isn't listed in ClinVar at all, and it's not listed in DECIPER either in any patients. So the likelihood of this being more than a kind of moderate level VUS at this stage is very low. And unless our patient's got a Barn-Dor diagnosis of Noonan syndrome, in which case perhaps some more assessment might be warranted, we can move on from here. So could we have the next slide, please? I think this is one of the trickier criteria that we use, and I think it's probably fair to say it's not one that we would necessarily always use as a standalone criteria, but one which certainly will make it much less likely in most circumstances that a variant is going to be pathogenic or going to be reportable. And this is when a variant is detected in an unaffected adult. And typically we'll find this is the case when we're doing trio sequencing, so trio whole exome or trio whole genome sequencing. And we would consider this generally relevant only for fully penetrant and relatively severe childhood onset disorders. So there's a significant caveat here and a significant amount of caution required around disorders which can be variably penetrant or expressive, and also around the possibility of low-level parental mosaicism being picked up in a trio, which I think is always something to be aware of in these circumstances. So the current NHS England guidance around whole genome sequencing is actually to dismiss what are the so-called tier 2 inherited variants in dominant genes, and these are variants that are detected in an unaffected adult in a dominantly inherited gene, which are not loss of function and which are not described as pathogenic in a couple of key curation resources. So these are variants that would typically be VUSs in most circumstances if you pick them up in a singleton, and obviously if they are also inherited in a dominant gene from an unaffected parent, they become even less likely to be pathogenic, and therefore our guidance is not to look at these in most circumstances. Again, I think there's some caution required here around disorders with known variable penetrance or incomplete or variable expressivity. I've put down some examples. There's by no means an exhaustive list, but the rasopathies, which I mentioned before, certainly one. A number of connective tissue disorders and several of the genes causing various forms of epilepsy are sort of known sources of pitfalls within genomic testing for this sort of approach. You can see here a really good really good cautionary tale on the bottom right-hand corner there, a box. This is a genuine case that I was involved with, I think more than 10 years ago now, a newborn who we did singleton whole exome sequencing with quite a florid phenotype really of scalpoplasia, unilateral eyelid coloboma, clefting requiring surgical reconstruction, bilateral talipes, and a cardiac anomaly. And this patient had a pathogenic variant, a very Barn-Dor pathogenic variant, in fact, in the gene COL3A1, which causes a form of Ehlers-Danlos syndrome, but one which is quite variably expressive because, as you can see from the pedigree here, this was inherited from the, at the time, apparently completely unaffected 21-year-old mother who was also heterozygous for this variant. So had we done a completely naive exome or genome trio in this patient and had rejected all variants inherited from an unaffected adult in a dominant form, we would have not seen this variant and we would have not made this relatively straightforward diagnosis. So I think caution around that approach, but I think still also a really useful annotation and a really useful quick way to give you an assessment that a variant is unlikely to be pathogenic in most cases. And in our hands, we would generally use this as a sort of supporting criterion alongside other clear pathogenicity flags. So we would have a rapid check to see whether the variant been seen in NOMAD, whether it has any apparent loss of function impact, so whether the PVS1 criteria would be applicable, and whether it's present in ClinVar. And if the answer to all those things is essentially tending towards the benign side, then the fact that it's been detected in an unaffected adult also will lead you to a point at which you can discard that variant. So next slide, please. The fourth criterion we use is where the variant type is clearly and definitively inconsistent with the disease mechanism. Again, in very small numbers of cases, this can be pretty much a standalone criteria for rejecting a variant, but we need to be assured that the mechanism is very clearly defined and that we understand it quite well. So that will not be the case in many circumstances, but again, it's often a really useful supportive criteria if you think your nomad frequency is likely to be too high for the disorder and there are no other clear pathogenicity flags. This can maybe be the clincher that gets you to relatively quickly to the point of dismissing a variant. And here I've shown three different circumstances where that can be relatively easily inferred from the protein genomic view in DECIPER, showing three different disease mechanisms and three different genes. So the top one is IFIH1, which causes a dominant ICARD syndrome phenotype. And it's well established that the disease-causing variants in this gene are the little yellow filled triangles there, which are missense variants in a couple of hotspots throughout the protein, and that the loss of function variants, so you can see probably it's a bit of a squint, but you can see just above the multicolored protein domain track, there are little red dashes, and these are all loss of function variants or truncating variants in nomad. And you can see that there's a significant number of them throughout the gene, including a small number of homozygous ones. You can also see from the predictive scores on the right-hand side, they're all cold colors and low numbers, so there's no constraint against loss of function in the nomad data set for this gene. We know in fact that loss of function variants or premature truncating variants in this gene are completely benign, and that missense variants are the only thing that cause disease. So if you see a loss of function variant, a truncating variant in this gene, is going to be benign. In contrast, in the bottom left, APC, a gene that I'm sure if many of you do colorectal cancer work will be very familiar with. This is quite a large gene with a large disorganized final exon, and we know that variants in this region are only pathogenic essentially if they are truncating, and that there are no well-established, highly penetrant pathogenic missense variants in this gene at all. So again, missense variants can be excluded with very little workup. And finally, a slightly more unusual mechanism on the bottom right there, the NEFH gene, which is associated with neuropathy. And the slightly unusual mechanism for this gene, which you can see sort of on the far right of the protein workup here, these little red triangles with a line flying off into the 3' UTR. These are final exon frame shifts that lead to an extension of the reading frame. So that's the only established mechanism within that gene. And again, other types of variants earlier on in that protein are not going to get above a VUS with our current understanding of that gene. So if we move on to the next slide, please. And I think this is the final criteria and probably one of the trickier ones. It's more of a combined criteria, if you like. These are variants where there's very little evidence in the initially accessible public domain data to favour pathogenicity, other than the fact that the variant would be rare enough to be potentially causative within that gene. So I think this is quite relevant in singletons and duos where you don't have parental inheritance data, and it's something again that can be very easily assessed with a small number of clicks using either the DECIPER interface, which I've talked about extensively, or key annotations from your NGS data pipeline. So the example I've got here again is a variant in PTPN11. So you can log in immediately, make your variant in DECIPER, and you can see that there are no identical DECIPER patients within that variant that have that variant, sorry. You can see on the right-hand side that it's been reported once to ClinVar, but with a variant of uncertain significance classification. So it's relatively unlikely that the PS4, PS2, PM6 criteria are applicable from that evidence. If your patient's phenotype is not a particularly strong match for Noonan syndrome or another associated rasopathy, then PP4 is unlikely. We can see that in the bottom left-hand side there. In the middle, we can see that the patient, this variant has 4 heterozygotes in the nomad V2, V3 data, considerably more if you link out to V4. So the PM2 criterion is probably quite arguable in this case with a Noonan syndrome, even though it's variably expressive. And you can see on the right-hand side from the protein view that the variant isn't in a hotspot, and there is a very clear hotspot nearby for PTPN11 pathogenic variants, but this variant very much is not in it. So the PM1 and PP2 criteria are quite likely here for this variant as well. So at this point, I think we can say this variant is highly likely to be not of clinical relevance. And I've put a couple of extra notes at the bottom of this slide as well regarding some of the other tools that one might be tempted to link out to in this case. So HGMD, the Human Gene Mutation Database, the HGMD Pro interface in particular, has lots of great links out to papers on a variant level. But actually, from a small audit we did in our laboratory some time ago, more than 80% of the variants that are in HGMD, the definitively disease-causing variants in HGMD, are now in ClinVar. It's probably more like 85% actually. And generally, if there aren't any flags that would tend you towards pathogenicity from your initial assessment, it's relatively unlikely that you're going to find anything with a deeper literature search. Potentially you can do this a little quicker if you embed a flat file from the HGMD Pro output, which is something that we're lucky to have access to in the UK, and certainly something that we do within our pipeline, or consider other paper scraping resources. So there are a couple of third-party resources out there that I probably won't labour the point on, that will allow you to pull out instances of a variant in papers and or doing Google searches. So these are all things you can do, but in this case with this variant, I think it's something I would not be likely to spend time on. And also in silico tools, the tools that allow us to apply the PP3 criterion. So using the ACGS 2024 guidelines, that would be REVEL for protein predictions or Splice AI for splice predictions. In most cases, when your variant is looking like it's not going to be more than a weak variant of uncertain significance, these aren't going to change your assessment. They're not going to get you from a weak VUS to a reportable VUS or to a likely pathogenic variant. And so actually looking at them is not time that is adding value to your analysis. So I would only use those in circumstances where we felt that the variant has got something going for it. So next slide, please. So I suppose, how do we apply all this in practice? Well, I think you need essentially a clear plan of the route that you're going to take for an assessment and how you parsimoniously record that outcome and that interpretation and make that available within your paper trail for your patient, so that it's there and recorded permanently, and for any assessors or any authorizers who see that analysis. And I think the most desirable approach for that is to have all of these annotations visible and readily accessible from your pipeline outputs. Sometimes that's not possible if your pipeline doesn't support that, or for example, in the England, the WGS outputs that we have are centralized, and we have the ability to link out to these resources, but they're not actually embedded within the annotations that we initially assess. So what I've put here is a little schema of the key annotations that I think we want to look at in order, and the source for those annotations in practice. So the first, clearly the most important, is what disorders and what inheritance pattern is linked to your gene. We get that either from our pipeline annotation or from the gene clinical tab on DECIPER. Inheritance info from our parents, if we have a trio, is probably the next most important item. Again, accessible from our pipeline output. We talked quite a lot about nomad frequency, but also not just the overall frequency of a variant, but the number of times it's been seen in heterozygosity, homozygosity, or hemizygosity in the case of X-linked variant, very important. And again, we get those from our pipeline or very quickly from the annotation allele frequency tab in DECIPER, which I think I showed you. The clinical case evidence, which we can get from ClinVar first and foremost, which can be, again, from our pipeline annotation, which can include our local databases or other databases of clinical curation and the number of times that the variant's been seen in affected patients, or more rapidly, I think, from the DECIPER summary and the annotation ClinVar tab, and finally the disease mechanism and mutation spectrum, which again is visible from that really nice protein genomic tab that I showed you, that will show you all the pathogenic variants in a gene that have been reported to ClinVar and DECIPER. And also the predictive scores for the gene, which demonstrate the constraint levels for the gene. So if we've gone through all those things, and in most cases that will take probably less than a minute, I think, for most genes, for most variants, and we still can't dismiss our variant as being unlikely relevant to the clinical presentation. At that point, it would be when we would do our full ACMG workup, and to consider looking at other sources as well, so things like in silico evidence and resources such as HGMD. Next slide, please. So in our hands, certainly in the southwest of England, we find that the majority of variants that are presented by our pipeline can therefore be dismissed from clinical relevance without having to do a full workup, and generally relatively quickly. Again, having audited this, it's around about 2% of the variants that are returned by our WGS pipelines where we would do a full ACMG workup. And I would say, again, really to reiterate that the ACMG criteria and a full coding workup isn't essential, nor is it desirable when it doesn't add value, and that is supported in the UK by our ACGS best practice guidelines for variant classification. It's essential that clinical scientists use their professional judgment for variant classification, and many variants prioritised by a bioinformatic pipeline will not require formal classification. Clinical scientists must identify those variants that require a full classification and then collate and critically appraise evidence for or against pathogenicity in the context of the individual case under consideration. For us, a key part of this is how we record that evidence and how we signpost the key criteria that's been used to dismiss the variant. You can see some examples of this down below here. This is a potted in a section of an analysis that was done on a whole genome sequencing case in our region. You can see the first variant is in the gene SAL4. It's associated with a skeletal dysplasia phenotype alone. This is, I think, was a patient with a syndromic intellectual disability that didn't fit the phenotype that was stated. And also you can see it has low level frequency in NOMAD, so that was dismissed relatively quickly with that analyst comment. The second variant is one that's associated most commonly with a recessive, listen carefully, phenotype, but also a more variably expressive epilepsy phenotype dominantly. This is a variant that had relatively high frequency in NOMAD, which would probably exclude a dominant causation, and was only recorded as VUS, and ClinVar had no other evidence to support it, so it was relatively quickly dismissed. The variant below is in a gene where there is only one recurrent pathogenic variant reported, so the variant was inconsistent with the established mechanism. Also, again, we can see there's low level NOMAD frequency for this variant. And the final variant on here was in a developmental disorder genes that would fit with the phenotype, but there was a single heterozygote in NOMAD. It wasn't in an obvious missense variant hotspot for that gene. It wasn't in ClinVar, and in this case, the analyst had also looked in HGMD quickly, determined that it wasn't in HGMD, so therefore this is going to be no more than a fairly lowly variant of uncertain significance, and no more workup really is required there. So all these four variants were dismissed relatively quickly without doing ACMG. Next slide, please. So a few principles I think that are important. Consistent approach by all analysts in terms of the extent of analysis done and how those decisions are recorded. And I think feedback from senior staff is really important. People respond very well to being told that actually you didn't need to do this bit of the job, that excessive analysis was done or excessive work's undertaken. And on the flip side of the coin, whether the assessment isn't sufficiently detailed or the outcome haven't been recorded clearly. And I think having really good training and competency materials will really help to guide and embed this approach in laboratories. And I think the way to do that actually is to focus the training on cases that are difficult. I think asking clinical scientists to do 10 normal cases under supervision before they're signed off for competency probably isn't a great approach. I think we need to focus the training on the tricky things and the difficult concepts, and also the pitfalls where you can come a cropper really with your pipeline, your system, or with the genomics of the disease in question. So any cases in particular from your clinical pathways where omissions were made in live analyses, and those learning points can be distilled and disseminated within your teams, really important to save those and embed those in your training. Anything where you've got something slightly unusual where perhaps the phenotypic assessment aspect, which I think is quite challenging in some cases, could be missed or misinterpreted in some way. And I've got an example to mention of that at the bottom there, which I think was quite a nice one that came up in our practice, where the variant fitted the phenotype, but actually was apparently not a great fit from the referral route to the panel. And this was a yellow box at the bottom here. This was a 15-year-old who was referred to our service with abnormal gait, worsened with cold and fatigue, the lower limb weakness and pain, and abnormal posturing. And this child came through a neurological referral route. So it was a neurology referral, and they requested, not unreasonably, given the phenotype, a dystonia panel, movement disorder panel, and a spastic paraplegia panel. So this patient was analysed on a WGS trio with both parents, and there was a variant which at first glance appeared not at all relevant phenotypically, a variant in the gene comp, which is a missense variant variant which had arisen de novo in this patient. And this gene causes completely non-neurological phenotypes. It's a skeletal dysplasia gene that causes two overlapping conditions, pseudoachondroplasia and multiple epiphyseal dysplasia. But actually, with a bit of further assessment of the phenotypes associated with those genes, we could see that they include hip osteoarthropathy, which can cause a waddling gait, childhood, potentially pain, and hip replacement in early adulthood. So it seemed to fit quite well actually with the gait features in this child, and a bit further assessment indicated this variant would have been a likely pathogenic variant using an ACMG workup, and it was reported back and turned out to be the diagnosis in this case. So it was a great learning case, I think, for our team there. I think another useful point to draw from your training cases is where variants are come across which may be unusually common in NOMAD in the population, but they underlie some sort of either incompletely penetrant or hypermorphic disease mechanism. So it's a couple of examples there, tyrosinase in oculocutaneous albinism and IFT140 in cystic kidney disease, where that's very much the case. And also considering any pitfalls of your pipeline or your decision support tool. So an example of that, which I think is quite relevant in the UK, is being aware of the fact that the NHS England whole genome sequencing pipeline currently doesn't co-tier, so it doesn't consider single nucleotide variants and copy number variants in recessive diseases. So the two areas have to be looked at separately for recessive diseases in the Genomics England analysis. And ultimately, I think what we get with that good training is we get well-trained, experienced primary analysts, and ultimately we can start moving towards a significant reduction in the amount of time spent authorizing or second checking effectively those results, and in time hopefully get away from that. So we have individual first analysts who are able to conduct an entire analysis and then only escalate to more senior staff for a sort of second opinion where there are difficult or complex cases that need or variants that need sort of additional input. Next slide, please. I want to quickly mention CNVs. I appreciate time is running a little short, so I will try and go through the remaining slides relatively quickly. So very much a similar approach to the SNVs, I think, for sequence-derived CNVs. We use DECIPER very much for this, for our whole genome sequencing cases, where we're looking at more of a gene agnostic whole genome assessment of CNVs. And we quickly record and dismiss CNVs where either the frequency, the disease association, or the impact on the transcripts excludes pathogenicity. Big fan of the DECIPER genome browser, which you can see here. So we have embedded a link out from our CNVs to be able to view this in the DECIPER genome browser. You can see, for example, this was a duplication which overlaps two different coding genes. But you can see from the way we have our tracks laid out, we have our omin morbid genes at the top, which show that there isn't a disease association for either of these genes. We can see that we've got population data from DGV Gold, which includes high quality normal arrays, the NOMAD structural variants track, which includes around 64,000 normal whole genome sequencing cases with high resolution for small variants, and then the NOMAD CNV track, which includes nearly 500,000 normal whole exome sequencing cases with the CNV encompasses 3 or more coding exons. So you can see here significant overlap for this duplication with duplications in those population control cohorts. We have the ClinGen dosage sensitive region track on, so we can see that this doesn't overlap any known dosage sensitive loci curated by ClinGen, and we can see the transcripts also of the just using the main select transcript for those two genes. So we can see that the likelihood of this duplication is that it wouldn't interrupt the reading frame of either. So we can assess this one as a benign CNV relatively quickly. Next slide, please. Probably won't labour this slide particularly lengthily, but I'm aware that many labs do confirmation of NGS and WGS results by an orthogonal method. This isn't something that we do routinely in our laboratory, again using the validation of the method, the technical validation, and the assurance of the technical pathway, ensuring that we are very aware from our validation work that sample errors, sample mix-ups are vanishingly unlikely in our pathways, embedding checks to ensure that's not the case. So things like checking the sex of patients, checking the relatedness of trios using SNPs, ensuring that the variant call meets our validated quality parameters. So when we do our validation, ensuring that we have a cutoff, a threshold for VCF quality score depth and considering also things like allelic balance and strand bias. And finally, before we report any SNV, and again, this is embedded within our NHSE guidelines for rare disease genomic testing, interpretation and reporting, which I'll call the INR guidelines for brevity. We would always view that the raw data on the BAM or CRAM file to ensure that there is no reason to suspect that the variant isn't real. You see on the left-hand side here an SNV, which is present on both forward and reverse strands at a good 50-50 allelic balance and a good depth. And on the right, a genomic deletion, which is in the top CRAM track and in the bottom one as well. And you can see here large insert reads that span the deletion range. You see clear deletion breakpoints that are delineated by the soft clip reads that are shown there by those red arrows. So these are variants that we would not confirm. Next slide, please. Talk a little bit about coverage and sensitivity. So horizontal exonic coverage is often used as a proxy for test sensitivity, but I think it's probably not a great one. We're aware there are variant hot and cold spots. We're aware that genes differ hugely in diagnostic yield and that there are various different variant disease mechanisms associated with multiple genes. So I think we need a pragmatic approach to assessing sensitivity. We do not need 100% coverage at your given cutoff for your gene panel for it to be a clinically appropriate panel. So when we're assessing sensitivity using exonic coverage, we need to think about any granular assessment that we do, any time that we spend looking at gaps in our coverage needs to be very much focused on frequent genes that are likely to be phenotypically key for that patient. So not rare genes, don't worry about those so much. Don't worry about genes that don't overlap the patient's phenotype. You need to be aware that NGS gaps are systematic and recurrent, and they can be dealt with generally by standard rules. Gaps in recessive genes tend to be lower risk than those in dominant genes because you're less likely to have two variants than one arising in your NGS gap, and variants can often be called with high sensitivity by pipelines lower than whatever your sensitivity cutoff may be, and actually visual assessment of those reads can assure you that your potentially suboptimal coverage region is quite low risk. Next slide, please. So just a couple of really quick examples here of establishing straightforward and evidence-based rules for acceptance of lower coverage in genomic assays. So PTPN11 here is a gene I've talked about a few times. You can see from the table on the right hand side, it's a very common cause of Noonan syndrome, attributing around 50% of the mutation spectrum in Noonan syndrome patients. But if you had a gap in exon 14 here from the image on the left hand side, you can see that actually potentially this wouldn't really be hugely concerning because it does not overlap any of the main mutational hotspots within the PTPN11 gene. And in fact, there are no ClinVar established pathogenic or likely pathogenic variants in exon 14. So you may accept that a coverage gap in exon 14 in your assay that's systematic is not a concern within this panel. Next slide, please. And likewise, thinking about long QT syndrome, this is a cardiac disorder where over 95% of the known cases are caused by variants in one of three genes: KCNQ1, KCNH2, and SCN5A. And you can see from the two images on the left-hand side there that the variant spectrum in KCNQ1 and KCNH2 is actually throughout the gene. So you would be very, very keen, I think, to have 100% comprehensive exonic coverage of both of these genes in your long QT panel. But then looking at all the other genes that have been associated with long QT, the table on the right-hand side, nearly every other gene will, if they have gaps in them, actually not reduce your sensitivity that greatly, because the percentage of long QT syndrome that's attributable to those genes, unless a patient has a very specific phenotype that links to one of those genes, is really, really low. So the likelihood is that any coverage gaps in those genes are not going to impact your sensitivity a great deal. Next slide, please. Reporting, I think, should be standardized. Generate your templates via LIMS. And I think quite importantly is assigning appropriate reporting processes to appropriate levels. And this was a slide I borrowed from Sean Allard here around potentially that a clinical scientist, a trained clinical scientist, does not need to be writing out no diagnosis reports or very standardized templated reports for established variant pairs that are reported as per ACGS guidelines and reported on template reports. It's possible to use other staff. Certainly in our laboratory, we have genetic technology staff that do this work to get these reports out and save your clinical scientist time doing this work. Next slide, please. Finally, I think I want to just make a quick point about MDTs and the use of MDTs in genomic testing. So what an MDT isn't for, well, it's not for pushing the responsibility for making a decision whether a variant is pathogenic or a VUS to clinicians. I've used the phrase untrained staff here, which is slightly facetious, but actually clinical scientists are the staff who are trained and accredited to make decisions around whether a variant is pathogenic or a VUS, and they should be the staff who are making those decisions. An MDT isn't reassurance, it's not a safety blanket, it's not a place for demonstrating how clever we are with regard to the variants that we've detected. It's not also there for explaining the results. The reports should stand alone and they may be accessed by other clinical staff in future. So what an MDT should be is a very focused query regarding the potential for a phenotypic specificity of a variant and or the fit of that variant to the patient, which will definitely influence outcome. So where a variant is on the VUS, likely pathogenic border, or where there's a decision between reporting or not reporting a variant. And it's potentially also a good area for discussing incidental or complex off-target findings following our national guidelines with support of our clinical genetics teams. And in our hands, we find that the vast majority of this work can actually be done by email, and that the number of in-person MDTs we do discussing variants is extremely small. And so next slide, please. And I think that the pie chart up here on the right-hand side just outlines that point. For our whole genome sequencing work in our region, we take around 2% of cases to MDT, and in almost all of those cases, those are conducted by email, and it's a focused discussion generally around the phenotypic fit or specific phenotypic features of the patient and at what level certain ACMG criteria relating to that can be applied. So they would generally be the de novo and the phenotype specificity criteria. It gives our scientists more time for focused analysis, and then we do alternative MDTs, which are not case-specific MDTs, to discuss our sort of educational strategies and do training with our clinical teams. So really much more streamlined way of doing it, I think, than having multiple in-person MDTs operating. Next slide, please. So I will probably not go into any details on this slide, just to make a plea that people follow the ACGS guidance, which is essentially that VUSs should only be reported where there's a high level of supporting evidence, and that where additional evidence might be obtained to allow reclassification as likely pathogenic. So these would generally be strong VUSs and or VUSs where we think that an additional action is necessary or is possible within that family to raise the variant to a likely pathogenic status. Next slide, please. And that's very much supported by our ACGS best practice guidelines, whereby VUSs should only be reported in exceptional circumstances. And again, in our hands, the number of VUSs we report is around 3 to 4% of our cases, so really relatively small numbers there. Next slide, please. I will also make a plea, I think, for laboratories to follow our BSGM guidance for incidental findings. So we know that pathogenic variants in highly penetrant genes where there is available treatment would be appropriate to report as incidental findings, but we would suggest reducing the number of incidental findings generally that are reported, not reporting likely pathogenic findings. We know from UK Biobank data, as you can see on the lower left-hand side here, that the penetrance of these has been shown to be considerably lower in the context of cardiomyopathy. And we're also keen, I think, that MDT discussion is most appropriately taken with expert clinicians who are out with the care of the patient or the family. It's very difficult once you've disclosed a variant to the clinician who is actually dealing with that case for them to then subsequently suppress it and not return it to the family. So having really good clinical genetics colleagues who are able to make value judgments in this space is incredibly valuable, we found. Next slide, please. I think this is sort of the final slide relating to sort of our approach here. So just demonstrating that at least within our region, within our laboratory, applying some of these aspects to our analysis workflow has really reduced the amount of time it's taking us to analyze cases, and we've seen the number of minutes it's taken per whole genome sequencing case in our lab reduce year on year from 2022, and we started doing a lot of sort of high volume WGS work to 2024, and also considering who does which aspects. So for example, as I mentioned earlier, having our technical team reporting out a lot of our normal and templated reports saves our clinicians a lot of time, our clinical scientists a lot of time, and enables us to comply with our turnaround time standards in the vast majority of cases. So next slide, please. very finally, I just wanted to put this up. It's very much hot off the press. I think this came out about 2 days ago. So we have a position statement from the ACGS in the UK, which many of my colleagues on here are co-authors. Very much recommend people read and absorb this statement. There are 19 explicit recommendations regarding rare disease genomic testing in the UK and Ireland, promoting timely and equitable access. These are across 8 different themes, and what I've covered today I think is largely the themes 3 and 4, which are noted down below. So I think very much would recommend that you have a read through that document. Next slide, please. So a very quick summary to finish with. All analytical processes should add value, so should either add sensitivity, accuracy or quality. Otherwise, in most cases, we can dispense with them. Not all variants require ACMG workup. Simplify your variant triage, so make sure you're concentrating on the criteria that will exclude a variant from clinical actionability. Ensure your key annotations are accessible. Continuous training and learning is important. Reviewing your process, focus on your errors, any omissions in your process and challenging aspects, and then really embed these in your training process. Ensure you're feeding back to your analysts that less is often more. Streamline your MDT process, use emails, use focus queries. Follow the guidelines that I've mentioned, the 2024 ACGS variant interpretation guidelines, BSGM incidental findings, and the NHSE interpretation reporting guidance. Ensure you have the right person in the right place, accessing your clinical pathways at the right time and doing the right jobs. Measure the impact of what you're doing, and last and by no means least, enjoy yourself, because you're doing fascinating and valuable work. And I do apologise, Sandy, for going almost over time.
Not a problem at all, Ian. I mean, that is a huge, vast amount of useful advice and information that I hope everyone really values and will put a lot of your key points and directions into practice locally. We do have a couple of questions. I know we're nearly on the hour, but I think it would be worth just addressing some of them. You say it was great to see actually the impact that you can measure in your lab. Has it been easy to implement? Do people accept the fact that everything's not going to be double checked? They're not going to rely on someone else just to check that your data or your interpretation of the data is accurate?
Yeah, I think the buy-in's been reasonably effective at most levels. I think a lot of this work has been done with our whole genome sequencing pathway, which is something that is relatively new anyway. So it's something that we've only been doing at high volume for about 3 years. So in many ways, when you're establishing a new pathway, I think establishing principles around that is more straightforward. But certainly within our NGS work, I think we've been working like this to some extent for quite a while now. So a lot of it is just embedding these principles and ensuring that they're staff are well trained, staff are receptive to that. But generally, if you're telling people actually they need to do less, it's a relatively easy sell, I would say.
That's a good
spend
way
more time on the interesting things and less time on the boring things.
And we all like that. That's a good way of putting it. Absolutely. I was really interested to see your lovely example of phenotype could be not quite as accurate when it comes to the gene with your pathogenic variants. I guess we're all very reliant on how good the phenotypic information is that comes with referrals. I guess that's where your MDTs can come into their own. You can have that discussion. And I know you mentioned it's very rarely you will sit round the traditional table to have an MDT. It can be an email, it can be a quick conversation, as long as it's recorded, I guess. Dave, I was going to come to you and just say this is the feedback we've been getting about how should MDTs be run, and I know we did a pilot last year. Any thoughts or feedback from the learnings from that pilot?
Yeah, I mean, it was relatively small scale last year. We're running it again this year as another pilot, just a bit of a plug. If there's still time to participate if you want to. I think, we've gone into it choosing scenarios that we think people will take to MDT. So, with input from clinicians. And I think, we could do it in any way. You can submit your results as, it could be an email discussion like Ian says, it could be a telephone call, it could be a meeting if that's what you would actually have in real life. And you can submit the results however you like for that EQA. So generally the way that you record your results. And we did find that worked. So we're looking at the overall scenario and where people go with that rather than how you actually do it, the level of detail you go into. So I think it's useful for that set of variants that would need to go into MDT.
Okay, thanks, Dave. We've got a couple of other, Ian, that's come questions on through the chat box. Do you think we should only analyse the genes listed on the test directory? And then there's a follow-up question from that, is also for those on the VCEP, so the ClinGen variant curation expert panel. What's your views on that?
So first question, I think my answer's probably no. I think we've made quite a few diagnoses, certainly in our hands, and I'm sure other labs that work in this space will have similar experiences of de novo variants and trios in particular, which are newer emerging genes. I think it's very much the diagnostic labs should be working in that space and should be collaborating with research on these new and emerging genes. With regard to the test directory, there's always an update process in that. So actually there are, an example this year, RNU42, probably the most common intellectual disability gene in man, is a new and emerging gene in inverted commas, but obviously we know if we're not looking at variants in that gene, we're potentially missing quite a significant number of diagnoses, and there is always going to be a curation period and a time frame whereby these new gene associations don't make their way onto panels. The panels are only as good as the curation. So
I guess
absolutely.
Back to your comments about using your own professional initiative and knowledge, isn't it?
Yeah, absolutely, and potentially having some guidance around actually how you do that and what sort of variants you would, what sort of off-panel variants you would spend a great deal of time looking at. Certainly not something we spend a lot of time with, but where you've got a strong candidate variant, I think there's very much benefit to doing a more appropriate workup with those variants. With regard to the VCEP guidance, I mean, it's maybe slightly off topic for this. I think probably fair to say I think some are better than others. Within the 2024 ACGS variant interpretation guidance, we suggested that labs essentially do their own sort of internal assessment as to the appropriateness of those guidelines when they're looking in their areas. I'm sure laboratories that work in specialist areas will be very familiar with the VCEP guidance within their own specialist areas and probably have their own opinions about the appropriateness of that. And again, I think it's completely reasonable for thinking from a very UK centric perspective, very reasonable for UK staff who have expert level knowledge in those areas to actually get involved in those VCEP opportunities and potentially to shape the way those guidelines are being produced.
Great, thank you, Ian, very much indeed. Emma, could you just pop up the next slide, please? Just finally to say, we've been some questions asking, will the slides be available? There's the acknowledgements for Ian, and then the next one, please. So yeah, we hold these webinars to address areas of interest and get some information out to our participants, really based on feedback. So please complete the survey that you'll be sent shortly. We do look at the feedback and we do change our approach accordingly. Lots of questions asked, will the webinar be available recorded? Future, you'll be emailed a link to the webinar after it's closed, and then also within a month, so in November, it'll be available on our GenQA YouTube channel, so please do disseminate that knowledge to others who may have missed the webinar today. You'll also get an automated participation certificate. That was another question that we've asked. And also, if you want to hear more about the educational events that we provide, then please sign up for our newsletter. So our next focus on topic, which is very soon, Dave, can you remember the date? It's definitely October.
Oh, I think it's the 15th.
So not long. So selecting tools for assessing and classifying genomic variants. So again, very pertinent to this audience. So please do sign up or contact us if you want any further information. So finally, a massive thanks, Ian, very much indeed. Huge amount of information there that hopefully people have found incredibly helpful. So thank you for your time today. And to Dave, Farah, Melody, and also Emma, who's been driving our webinar today. So thank you everyone very much, and hope to see you on the 15th on our next Focus On webinar. Thanks all. Bye-bye.